Hi, my name is Jessica Redline, and this is for LAPU Micro 225, lab number 8, and it is August 6, 2020. Today we're going to be doing the carbohydrate fermentation testing. So this is exercise 1. I have all my my phenol red tubes here, and they're all labeled and ready to go. I have them in this little stand here just to make it easier. I have my candle lit here, and I'm gonna pass the tube lip over the flame to sterilize them. I also have my Durham tubes that we're gonna be using for this experiment. I have my fructose powder, fructose, glucose, and my mannitol. I'm just gonna get my goggles on really quick. Okay, so now, so like I said, everything is labeled. So what we're gonna do is I have my pipettes with some alcohol in a cup. Sorry, my goggles are fogging up. I'm gonna grab number 1 of my Cervise. This is the glucose tube. I'm gonna pass it through the flame to sterilize it. So just carefully pass it through. Then I'm going to get my glucose, and I'm gonna pour half of the powder into the tube. Maybe a little bit more. Okay, I would say that's maybe a little bit more. Okay, I'm gonna close that back up. I'm gonna run through it again through the flame before I cap it. I'm gonna screw it on. And I'm going to shake it vigorously until the powder dissolves. I also have my Durham tubes that they've already dried in alcohol. I did that before, and then I just placed them back into the bag. They've already dried thoroughly. I'm gonna insert one of the Durham tubes where the opening is facing downward. So that one I shook pretty good. I'm gonna drop it in so it's sitting like that. And I'm gonna get another one. This is S. epidermidis, so we're just gonna keep repeating the steps. And I'm going to open this, pass it through the lip. Pour the rest of the glucose powder into this one. I'm gonna pass it through the lip again. Shake it. It's about dissolved now. I'm gonna drop one of the Durham tubes inside. Just like so. So that one's for the glucose, that one's done. We're gonna move on to number 2, a Cervise in fructose. Pass it through the flame. We're gonna pour about half of the fructose in here. And that's about half. I'm gonna cap it now, and I'm going to shake it. I think it's about dissolved. I'm gonna drop the tube into there. I'm gonna grab my nether fructose S. epidermidis. And we're gonna pour the remaining fructose in there. Last one is the mannitol. So number 3 for S. cerevisiae mannitol. Pour about half into okay, that's about half. No, I realized I just forgot to put a test tube in the other one. And the last one. So we'll do that now. Derm test tube into number 2. Continue shaking this one. I'm gonna add a derm test tube in this one. Just like so. Last one for S. epidermidis, number 3 for the mannitol. I'm gonna add the remainder in there. Shake it vigorously. And then we have our last test tube to add to this one for the derm test tube. So those are sitting nicely. And then it says to remove 1 pipette of alcohol from the alcohol cup and shake dry. So I'm gonna do that. And I'm gonna grab my live cultures, my S. cerevisiae and my S. epidermidis really quick. So for the S. cerevisiae, I have it here. This is my cultured broth. I'm going to pass it over the lip of the flame, just like so. And I'm gonna grab my pipette that's been dry, and I'm going to, after I pass this through the lip of the flame, I'm going to transfer 4 drops of the active culture to each of the phenol red tubes that are only labeled the S. cerevisiae. So, up here. S. epidermidis. This is S. cerevisiae. I'm just removing the caps as I go. 4 drops into here. Okay. I'm just gonna place the caps on real quick of those ones. And then my S Service A mannitol one. It's kind of hard to do this with. Going to grab 4 drops of that in here. Okay. I'm gonna cover this back up. I'm gonna place this used pipette into some bleach. Over to the side, I have some bleach. I'm gonna stick it in that. I'm gonna cover these ones that I just did of the S Service A that have the culture in them. Now, next one I'm gonna do is the S epidermidis. So let me unscrew the lids for this really quick, just to make it easier. Okay, so I have them all setting up. Now I'm gonna grab a new pipette that's in alcohol and shake it dry. I'm gonna get my S epidermidis culture. And I'm going to place, I'm gonna actually pass it over the flame. And I'm going to draw out the liquid and place 4 drops into each one. Not touching the top of the lip with the pipette. And I'm gonna place this pipette also in some bleach, diluted bleach. Now it says after I've repeated those steps, I'm gonna put the caps back on, and I'm going to return these cultures back to the fridge for further uses on projects. And I'm going to incubate these red baths for 12 hours. So then I will update you on the result and what I find on what happens with these when they sit for 12 hours to ferment. So I will return that with the results video. Thank you.